Welcome everybody to today's Natural History Live. I'd like to pass over to our speaker for today, Dr. Tammy Shadbolt from ZSL, who'll be talking to us all about protecting the health of Britain's rarest wildlife.
Thank you very much, Beck. Okay, let's get started with today's talk. So a few signposts for you to start with to guide you through this presentation. A brief introduction, and then I'd like to cover three particular things. That is the principles of disease risk analysis in conservation, disease risk management during translocations, and the post-release health surveillance for rare species. And then I'll just conclude with some thoughts on future directions. So a short introduction. Brief background, how I came to be involved with wildlife conservation. So I began my career as a small animal general practice vet. I worked in the UK and internationally in about 7 other countries in both charity and private practice. And I moved into teaching when I came to London in about 2012-13, gained a PGCert in veterinary education, and I continued to teach as a teaching fellow and examiner for the Royal Veterinary College. I gained my specific training in wildlife health and research through an intercalated BSc, an MSc and PhD, and I continue to supervise research students, which I particularly enjoy. I'm fortunate now because my role at ZSL actually combines a lot of different things, so clinical work, teaching and research in wildlife. So I work as part of the Disease Risk Analysis and Health Surveillance team, otherwise known as DRAHS. And we're based at the Institute of Zoology, Zoological Society of London. And you might know us better for the running of London Zoo and Whipsnade Zoo, but we are heavily involved with conservation. So DRAHS is funded by Natural England, and that is the government's advisor for the natural environment in England. And importantly, we work with multiple wildlife organisations, groups, charities, as well as academic partners. So what do we actually do? Well, we carry out 3 main things, that is disease risk analysis, disease risk management, and post-release health surveillance whenever conservation translocations of rare native species are carried out in England. And those are the 3 elements that are the subject of this talk today. So to get started, we probably need to understand a few key concepts. And the first is, what are conservation translocations? Well, this refers to the intentional movement of animals from one area to another for the purpose of reintroduction, reinforcement, assisted colonization, or ecological replacement. Now, reintroduction is quite a familiar term, and it refers, of course, to the bringing back of animals to an area within the native range from which they have gone extinct. Reinforcement involves adding to a population within the native range, and that's usually done to increase numbers or perhaps genetic variability. The terms assisted colonization and ecological replacement you may or may not have come across. So assisted colonization involves the movement of animals outside of their native range to an area where they can better survive. An example would be, actually, we have some corals in the photo on the screen, and corals are quite frequently these days moved from degraded reefs to artificial reefs where they can flourish and we can conserve the species. So ecological replacement finally involves the moving of animals again outside the native range, but in order to carry out a very specific ecological role. So I suppose an example would be a bird species moved in order to facilitate seed dispersal in a particular environment. So due to human population expansion and development, wildlife habitats are in decline and species are too, unfortunately. In addition, some wildlife species are at risk of direct or indirect persecution still. and we'll come on to talk about that later when I go over one of the case studies. So conservation translocations are on the increase in order to counteract this, but they really do require multidisciplinary teams to carefully oversee the planning and implementation. So there's a number of risks, and that might include stakeholder risks, such as economic risks, livelihood or job risks if the translocations fail, ecological risks, which may refer to things like the collapse of food webs or altered habitats by the relocation of animals. And finally, disease risks, which are the focus of my particular team. Now, disease risks are inherent in the translocation of animals. And that's because when we move an animal, we're moving a biological package of organisms. So even you or I have numerous bacteria and parasites both within us and on us, as much as we may not want to think about that. And we come in contact with infectious agents every day. And as we know, if things change in our environment, if the weather patterns change, if we become very stressed, then our susceptibility to becoming diseased from those infectious agents does change. And it's the same for animals. And that's why we have to think about it during translocations. Okay, so I'm going to move on and talk through some of the principles of disease risk analysis in conservation. So first of all, what is disease risk analysis, or DRA for short? Well, DRA is a method for examining these risks of disease in a specific planned conservation translocation of animals. And carrying out a DRA is considered an essential planning stage of a conservation translocation, according to the IUCN guidelines, that's the International Union for Conservation of Nature. If you're not familiar with IUCN guidelines, then I really would encourage you to take a look at the array of publications they have on this subject on their website, and the address is shown on the screen, and I'm sure Beck will pop it in the chat as well for you. So what is the theory behind disease risk analysis? Well, it might be helpful to look at this diagram. So this is a translocation pathway. And if I can get the cursor working, I can talk you through it. So here we have a source population of animals. And in this example, we're going to translocate them via a lorry across a mountain range to the destination population B. Now, these yellow biohazard boxes represent points along the pathway where there may be a risk of disease. So in my team, we refer to these as source hazards, carrier hazards, transport hazards, destination hazards, and population hazards. But it's slightly outside the scope of this particular talk to go into that in great detail. To give you an example though, a source hazard would be an infectious agent that the animals at the original site may be carrying, and they may or not be diseased with that, hopefully not. But when they are translocated, there is a risk that those infectious agents could be released into the destination population. So we worry about source hazards. Transport hazards are another one that's quite easy to understand. You can imagine that the animals could pick up parasites or bacteria or infectious agents within the transport vehicle itself during the translocation. So essentially, and for the rest of this talk, when I talk about either ecological parasites or hazards or disease risks, I am referring to that array of infectious agents that includes bacteria, viruses, fungi, and your macro and micro parasites. So how is a disease risk analysis carried out in practice? Well, a DRA usually takes about 6 months, and it involves extensive literature review and the collating of expert opinion. So this is all compiled into a comprehensive qualitative research report, and it's used by our collaborators to decide if those risks that we have identified as possible during the conservation translocation are acceptable risks to take or not, and therefore whether the conservation translocation should or should not go ahead. So why is disease risk analysis important? Well, if you haven't analysed all those infectious agents that could cause problems along the way, then a number of things could happen that we really don't want to. So the first is that we may compromise the health and welfare of individual animals. Now, there's a picture of a rabbit on the screen which has myxomatosis disease. I'm sure you're familiar with it. And this is caused by myxoma virus, an infectious agent that was unfortunately intentionally introduced to a number of countries around the world. Now, myxomatosis unfortunately causes a long death and a lot of suffering. The animals tend to develop necrotizing and swellings, lesions around the genitalia and the ocular area, and it's not nice to see. The second thing that could happen is that disease may lead to death and actually reduce numbers of animals within a population. And for a rare species, that may severely restrict breeding success. It could create a genetic bottleneck, which is a term you may be familiar with, and it may even lead to extinction, especially through frequency-dependent disease dynamics, for those of you that are into epidemiology. Thirdly, disease does change the dynamics of the ecosystem, and we mentioned food webs previously. So I'm thinking something like the loss of a prey species could affect the food available for a predator species. So hopefully you can see why disease risks can have quite serious effects if we don't know about them. Right, onto disease risk management during translocations. Okay, what is DRM, or disease risk management? Well, DRM aims to mitigate disease risk by implementing practical action before and during a translocation in order to ensure animals are healthy. There's a number of practical steps I'm referring to when I say that, and there's a few images on the screen to exemplify what I mean. So if I just pull my cursor up again, here we have someone using hand sanitizer. Unfortunately, we're all very familiar with this now. It's really important to think about biosecurity when we are undertaking a translocation. We do not want to inadvertently move infectious agents between animal populations, either on our own hands or perhaps our clothes or other fomites. So we use hand sanitizer, we use disinfectant, we may wear overalls when we're working with these animals. The second term, again, unfortunately, we're all rather familiar with, quarantine. It refers for us to the keeping of animals in a biosecure facility pre-translocation in order that we can really do some health checks, keep an eye on them and check that disease doesn't emerge. The stethoscope here refers to the health checks that we do. So animals receiving full veterinary examinations. It's a really important part of the procedures of disease risk management. This image here refers to the number of disease screening tests that we can carry out during or before a translocation to check for any bacteria or parasites that animals might be carrying and could be a risk to that free-living destination population. This image here refers to treatment. Now, that could be prophylactic treatment, or it could be medicines to treat disease if we have found it during the screening or the health checks that we do on the animals. And finally, you're all familiar with this, it's a microchip. So I expect your dogs and cats are microchipped. We do try and microchip individuals that are part of translocation programs. Not every species can be microchipped, but we certainly do that. And that helps us follow the fate of individuals as they progress during the translocation and afterwards as well, which we'll come on to talk about later. So the reasons for carrying out disease risk management are numerous. It's imperative that we remain responsible and accountable for maintaining animal health and welfare at all costs during translocation. It's very important that animals that we are releasing are fit to withstand the stress of translocation and the change in their environment, but also so that they are fit to survive, to thrive, and to reproduce. Finally, it is essential, as we have already mentioned, not to inadvertently introduce infectious agents that could cause disease in wild populations, especially when that destination population might be naive. And what we mean by that term is that it may not have met certain infectious agents before, and that does make it particularly vulnerable to any new incoming infectious agents. Okay, so from here on in, I'm going to use some case studies to try and put what I'm talking about into some kind of context. So the first case study is the Hazel dormouse, very cute species and quite rare in England. So the translocation of captive bred Hazel dormice take place every year into protected wild woodland sites. Now, before they're released, these dormice come to us at ZSL and spend 8 weeks in quarantine with us, where we undertake really extensive checks, monitoring, and disease screening. So I'm going to play this video. It's 2 minutes long, and it's a nice way to summarize what we do to ensure disease risk management for the Hazel dormice. So hopefully this will play, and hopefully you'll be able to hear the sound at your end.
So we're here today to release the dormice that we've kept in quarantine at ZSL into soft release cages. It's a release program that has been led by PTS, the People's Trust for Endangered Species, and both ZSL and Painted Zoo, we keep about 20 dormice each year in quarantine that undergo health examinations and are then released at the release site. So they're being kept in big cages and one male and one female together. They're still being fed, and then at the end of the soft release period, the cages are opened, they can go into the woods, but food will still be supplied to them so that they can have some supplementary food if they're still finding it difficult to find food in the wild. So at ZSL, we've been involved with the dormice release program for many, many years, and our focus is health and disease. So what we're trying to do is to make sure that they're as healthy as possible when they're released, but we're also looking at the fact that we don't want them to inadvertently bring parasites or diseases with them into the wild, which they would be then introducing to this woods. So that's why we do health screening while they're in quarantine. We chip them, so we introduce a microchip under the skin between their shoulder blades when we're doing the health examination under anesthesia. And with that chip, each individual can be recognized with a chip reader. So this is a big day for us because it's a culmination of a period of about 2 months of keeping the dormice in quarantine, doing health examinations, doing stool checks. So it's quite a special day for us because it's the end of a busy period where we've been making sure that the dormice are healthy and fit for release.
Okay, so the importance of disease risk management in this project is probably emphasized if we think back to 2011, when we did detect a particular tapeworm whilst the dormice were in quarantine that we were worried about because we don't think it's out there in the free living population. And so the dormice were treated with a wormer a bit like your cats or dogs. And we continue to fecal screen the dormice about 3 times during their quarantine period, year on year. So the good news is that we've never picked up this particular parasite again. And further to that, the Hazel dormouse program is really going very well. So in 2021, we released the 1,000th dormouse from the program. And you might have seen quite a lot of media coverage from this in June, particularly put out by our partners at FHEA. Okay, so the second case study that I wanted to talk through with you is that of the checkered skipper butterfly. So checkered skipper butterflies became extinct in England in 1976. And genetic evidence interestingly showed that Belgian butterflies were evolutionarily similar to those that had existed in England. And so translocations to England began in about 2018. Now, DRAHS vets carry out pre-transport and pre-release health checks of these butterflies. So here's some pictures for you to illustrate the case study. Here we have on the left a checkered skipper butterfly in a collection pot, and we are examining it. We can take body measurements, even body weight within the collection pot prior to transport to England from Belgium. In this second picture, we have the butterflies released into this mesh tent, and this is at the release site. And it's a good opportunity for them to adapt for a few hours to their new environment and for us to observe that they're doing okay. And we can do further health checks at this point if we need to. Last but not least, we have a healthy checkered skipper butterfly, which has been released. Now, the Checkered Skipper program is also going very well. I'm pleased to say we have not detected any infectious agents in the new population in England. And we also know that this English population is now breeding successfully. So the Checkered Skipper is back in English woodland, which is fantastic. Okay, so for the final section of my talk today, I want to go through post-release health surveillance for rare species before I wrap up with some thoughts on future directions. So what is PRHS, or post-release health surveillance? Well, this involves monitoring the health of free living populations of animals in order to rapidly detect disease threats. And we can do this in many ways. The 2 ways which we use most of the time are PRHS in the field, so going back into the sites where the animals have been released and carrying out health checks on the animals that we find. Now, hopefully, we'll be able to identify individuals that have been released and check those in particular. But it's also really important to carry out health checks on the free living population, so those that are breeding successfully in that wild site. And also the species that may live there as well that are quite similar, because they could harbour similar infectious agents. The second thing that we do is post-mortem examination on animals found dead. So this allows us to not only examine the animals that have died, but also to carry out a range of tests. So I tend to say to my students, think of all the ologies. So radiography, which is slightly different, I suppose, but then bacteriology, mycology, virology, histopathology, and toxicology. And the reports that we write using the results of our findings, they go to our submitters, to our collaborators, and they go to Natural England. So why is post-release health surveillance important? Well, we want to ensure, of course, that we have released healthy animals, those that are fit to survive and thrive and reproduce in the wild. And the information we gain from PRHS may help us identify ways that we need to change the disease risk management. So it's an iterative process, and that's really important. The other thing is that we want to build up baseline data about these rare free living species. Unlike domestic animals, there's quite a lot we don't know about wildlife species in terms of anatomy, physiology, and the infectious agents that they carry. We also want to find out, of course, what the contributors to mortality are in animals that have been found dead. And note that I use the term contributors to mortality. We quite rarely come up with an exact diagnosis of death. And that is because wildlife is submitted as fairly decomposed carcasses, I'll be honest, and also because we don't have any ante-mortem clinical history. So we don't have clinical signs and information about what was going on for that animal prior to death. So it takes quite a lot of detective work to find out what might have contributed to its demise. Finally, of course, we really want to be aware and detect new and emerging disease threats in wildlife. And this is probably one of the most important aspects of PRHS in veterinary conservation medicine, because the animals we're talking about are inherently vulnerable as a rare species. So a couple of case studies for you, again, just to put some things in context. So the first is the pool frog. Now, the pool frog was reintroduced in 2003, initially from Sweden, and there have been multiple translocations carried out between the two created populations in England via head starting, which is a protective captive rearing method, which require another talk to go into in depth. Now, DRAHS vets carry out annual health checks on the pool frog populations in the field. And that includes free-living metamorphs and adults, and in addition, common frogs and common toads as well. So as we mentioned earlier, it's important to check the health of animals that live with the species of concern. Of particular importance in amphibians, we swab all our pool frogs and common frogs and toads that we survey for BD infection. So we take swabs and we do PCRs in our laboratories here for that. Now, you've probably heard of BD infection. It causes chytridiomycosis, which is a horrible fungal disease in amphibians now worldwide and does cause death. So pictures, we have a pristine pond at one of the release sites here on the left. In the center is one of my veterinary students, and you can tell that she's wearing quite extensive PPE, and that is, as we said earlier, so that we don't spread any infectious agents between populations. She's actually carrying out a check on the heart rate of this pool frog using a Doppler ultrasound probe. It's really effective for those of you that are vets on the call. So it's really good to hear the heart using that, and better than a stethoscope actually. And finally, we have a healthy pool frog undergoing examination here. Now, thanks to, I think in part, the rigorous PRHS that's carried out, the pool frog program is also doing really well. We have fairly well established populations. No BD or infectious agents have been detected in the free-living pool frogs in England. My final case study of the day is the red kite. Red kites were introduced in the 1990s to England, and translocations are no longer carried out. You'll probably understand why if you've ever driven the M40 corridor. You'll have noted the birds circling overhead, and the population is really thriving. We think we've got probably over 5,000 breeding pairs now. So DRAHS vets monitor the health of the red kite population by carrying out post-mortem examinations on birds that are found dead and submitted to us. And we carry out a lot of testing to try and understand the threats that this population may still face. A slight note, the next slide, and only the next slide, contains images of a dead bird, so please feel free to look away if you would rather not see this. So the picture on the left is of a healthy, beautiful red kite in flight. The picture in the middle of the screen is a radiograph of a dead red kite. And you'll see that the red arrow is pointing to a fracture through the radius and ulna of this bird, or the wing bones or the arm bones of the bird. And those of you from a veterinary background or discerning eyes will see that there is also a fracture through the tibia tarsus, so the leg bones of this bird. This is quite a classic presentation for impact trauma. So we do get a number of red kites that have been found on grassy verges by main roads, and they are submitted to us, and most likely they have been hit by vehicles. Now, the picture on the right is a transection through the heart of a red kite, and hopefully you'll be able to see there is a round metal object in the heart muscle. And that, unfortunately, is a shotgun pellet. So unfortunately, that is the consequences of the illegal shooting that still does take place in this country, even though these are protected birds. Finally, I haven't included a picture of a red kite with internal hemorrhage because it's not very nice to look at. But the consequences of second-generation anticoagulant rodenticide poisoning is something that we do see very regularly. The reason I've actually used the red kite as our last case study today is to highlight that although we focus on infectious disease a lot of the time, we do also keep a lookout for non-infectious disease threats. And I'm afraid that non-infectious disease threats, often as a result of human activity or even persecution, do still continue to threaten the wildlife populations in this country. But let's not end on that slightly downbeat note about the perils facing red kites, and rather some positive thoughts on future directions. So DRAHS will continue, as we always have done, to carry out disease risk analysis, disease risk management, and post-release health surveillance for new conservation translocations that occur within England. We are constantly evolving our collaborations with conservation organisations so that we can help them maintain healthy wildlife populations. We continue to research new methods for assessing and detecting disease. And I particularly enjoy teaching the next generation of scientists. And most of us here at the Institute are involved with undergraduate or master's programs on wildlife health and wildlife biology. So I'd like to conclude today by saying thank you for attending, because your interest and involvement really does matter to all of us working to protect England's rare species. So please do get involved or continue to be involved with the fantastic conservation organisations in this country. And please do help us generate awareness of what our key messages would be, which are that conservation translocations are complex and they do need to be undertaken with a great deal of care. And if they are, then they can be very successful. Please do visit our web pages at the address on the screen, and I'm sure Beck will pop that in the chat too. And you'll find there links to project overviews, blogs, academic papers, as well as to the conservation organisations that we work with, including PTES, BC, that's Butterfly Conservation, or ARC, the Amphibian and Reptile Conservation Trust, who are involved in the case studies that I've mentioned today. So please do get in touch with us at DRAHS email as well, which is also on the screen. And in particular, if you'd like advice on best practice in disease risk analysis, disease risk management, or post-release health surveillance, we would be delighted to hear from you. Thank you very much for listening, and I'll take any questions.
Perfect. Thank you so much, Tammy. So we'll go to the floor first to see if anyone does have any questions. So if you'd like to put your hand up, your virtual hand, we can come to you. But we have got a couple of questions in the chat, so we'll start there. And if anyone does pop up, then we'll jump to there. So first question we've got is from Stephen, and they ask, do similar principles apply to reintroduction and reinforcement of plant populations?
I would say yes, because at the end of the day, they are living organisms, and therefore infectious disease can occur in living organisms, be it plant or animal. That's probably as far as I can go with that, given that I'm from a veterinary background and therefore specialise in animals.
Perfect. So that kind of links to a question that Diana had, who asked, do rescue centres have to take the same precautions as you do? I'm assuming they mean in regards to releases and things like that of animals.
Yes, so there is actually a whole sort of different bank of rules around release as part of rehabilitation. So certain species are not allowed to be released now into this country if they have gone to a rehabilitation unit. So I would direct you towards things like the RSPCA wildlife hospitals, and they would have the kind of associated rules with that. But certainly the same precautions need to be thought through, but there are different rules for different species.
Great. Okay, so another question we've got is, how much is the risk both to the individual organism and to an overall ecosystem over the use of disinfectants such as chemicals?
That's a good question. We obviously do use disinfectants to reduce the risk of infectious agents, of course, during translocation. Now, these days, disinfectants are getting better and better, and the important thing to do is do your research into which ones are okay for animals and to be used around animals. So, for example, here we tend to use Safe 4 as an example in our projects with animals. So it is important to think about disinfectants and certainly how those are discharged into watercourses. Watercourses are not something I specialise in, but certainly that's a topic in itself.
Yeah, I know when I've worked with animals, we've always used Safe 4. Safe 4 seems to be a very common one that a lot of places use because of it being a bit more environmentally friendly as well, isn't it?
Yeah.
Great. And last question that we've got is, do you have any ideas where the dormouse tapeworm came from?
No, in short. Essentially, it obviously came from, I guess, the captive breeding population. Now, we do keep in touch with the captive breeders group, and so results will be shared with them, and we sort of advise on checking the health of the animals at that stage as well, if that would be a sensible thing to do. But where it came from originally, I can't totally put my finger on, but certainly it was detected because of our daily health checks within quarantine here at ZSL.
Okay, great. And there's one more question. So can BD jump to newts as well? So obviously we find it in amphibians. Can it jump to newts too?
So BD and BSAL. So there's a new disease or version of BD, which generally affects salamanders. But yes, it is a problem in amphibians in general, BD. And BD and BSAL are swabbed for in most amphibians now, whenever we're out doing post-release health surveillance. It's really important when you're thinking about species that may have been known to harbour these things, to appreciate that infectious agents can jump species as well at different times. So we scan or we swab for these types of diseases all the time to check for that as well.
Okay, great. Yeah, no, it's definitely one of the ones that you hear a lot about, isn't it? It's one that's definitely affecting a lot of our native amphibian populations. Okay, well, that's all the questions from the chat. So if no one does have any further questions that they would like to ask, we'll give you a couple of seconds just to have a think, see if there are any further questions. But if there's not, then we'll pull this session to a close. And thank you everyone for attending, and to our speaker to today as well. Don't think there's any further questions.